All right, let's get started. Are there any questions from last time? Okay, so this chapter is about transcription and translation. So we want to see how you can go from DNA to protein, right? As I said last time, right, the information from DNA is first copied into RNA, right? And then after that, that is translated into protein, right? So during transcription, the cell copies the required part of the DNA sequence into a nucleotide sequence of RNA, right? And we know that DNA and RNA, they have similarities, right? But there are differences also, right? So what is the sugar in RNA as opposed to what in DNA? Yeah, good, okay. And then in DNA, what are the bases? Okay, and in RNA, the different U is there instead of T, okay? So keep that in mind, right? So to initiate transcription, a small portion of the DNA double helix is unwound, and one of the two strands of the DNA double helix will act as a template for RNA synthesis. Okay? So just like for DNA replication, we saw that the enzyme that carried out DNA replication was what? Not primase, DNA polymerase, yeah. So for RNA, for transcription, it's going to be RNA polymerase. Okay? So the RNA synthesis is carried out by an enzyme which is called RNA polymerase, which catalyzes the formation of an RNA strand that is complementary to the DNA template that you're looking at, that RNA polymerase is looking at, right? And the process is very similar to DNA replication by DNA polymerase. And again, this is going to use energy-rich nucleoside triphosphates. Okay, why? Because the reaction that you're trying to, the reaction that you're trying to make undergo, all right, that reaction is energetically unfavorable, all right. So you need to break up some bond somewhere, and you need to supply the energy, right. So that's why it'll start off with energy-rich nucleoside triphosphates, right, and then you'll get a monophosphate that will get absorbed into the RNA strand. Okay. So these triphosphates are hydrolyzed to drive the formation of the RNA strand. However, transcription and DNA replication, they have some major differences also, right? First of all, since RNA polymerase synthesizes RNA, it is going to catalyze the linkage of ribonucleotides. The sugar is going to be ribose, right? Instead of deoxyribonucleotides, right? Number one. And then the second thing is that unlike DNA polymerase, RNA polymerases do not possess any nucleolytic proofreading activity. Remember, I told you last time that DNA polymerase, before it adds a new nucleotide, it is going to go and check its work, right? And if there is a mistake, it will go and take out a nucleotide, right? So it has got nucleolytic activity in the 3' to 5' direction, right? RNA polymerase doesn't have any of that, right? So RNA polymerases do not possess nucleolytic proofreading activity, and as a result, RNA polymerases can start an RNA chain without the need for a primer. Okay, they can start from scratch. And of course, if they're not checking their work, the RNA polymerase is probably going to make more mistakes, right? But that's not, that crucial, because RNA is being produced on an as-needed basis. It's not the storehouse of information, right? So it doesn't have to be, produced that faithfully. Okay, any questions? So transcription is a lot less accurate than DNA replication. So if you have RNA polymerase that is transcribing, right, into RNA, it is making one error or one mistake for every 10,000 nucleotides that are copied, as compared to one error in every 10 million nucleotides that are copied in the case of DNA replication, right? Now, since the DNA serves as a permanent storage of information, while RNA is only a temporary intermediate, it is only imperative that DNA replication be much more accurate than transcription, right? Because otherwise everything will get messed up. Okay, any questions? Now in transcription, right, the newly formed RNA strand does not remain hydrogen bonded to the DNA template strand. If you remember when we were talking about DNA replication, right, DNA replication was semi-conservative, right? I mean, so in the new strand, in the new double-stranded DNA, one of the strands would be coming from the previous strand, right? And you would also get double-stranded DNA when the process was completed. In in RNA production, right, RNA polymerase is just going to produce single-stranded RNA, right? And so there is no hydrogen bonding with the original DNA strand, right? So instead, the DNA helix is going to reform, right, once the RNA has been copied out from the template, and it is going to displace the RNA chain. So as a result, RNA molecules that are produced by transcription are single-stranded, right? So that's another important difference between DNA and RNA, right? DNA occurs in nature as a double-stranded molecule, as the famous double helix. RNA occurs as a single-stranded molecule. Any questions? Also, RNA molecules tend to be much shorter than the DNA molecules because the DNA molecule is basically the entire genome, right? You'll have DNA molecules, but RNA is only the portion that needs to be translated into protein, right? So it's going to be a lot shorter. Also, the immediate release of the RNA strand from the DNA as it is synthesized means that many RNA copies can be made from the same gene in a relatively short period of time, right? So as soon as it is made, as soon as the RNA strand has been produced, it is released, right? That means the DNA is again available for making a new RNA strand, right? So things can move a lot faster. Now, so the process of going from DNA to RNA is referred to as transcription, right? And the process of going from RNA to protein, right, that is referred to as translation, right? Today we will finish up both transcription and translation. By the time we get done today, like you would have been given all the information that you need to know about transcription and translation, right? However, not all the RNAs that are produced in a cell are meant to be translated into protein, right? So there are three different groups of RNAs. The first one is called messenger RNAs or mRNAs, right? So the little m in front of RNAs means messenger RNAs. These are the RNAs that direct the synthesis of proteins. Okay, these are the ones that will get translated into proteins, right? Then in addition to that, you have two other classes of RNAs, and we will see their function, before this chapter is over. The first one, these are called ribosomal RNAs or little r RNAs, right? They form the core of the ribosomes on which mRNAs are translated into proteins, right? Ribosomes are basically the molecular machines that make proteins, right? That basically take in the messenger RNA and will produce a full-fledged protein corresponding to that messenger RNA, okay? And I will show you how that is done, right? So ribosomal RNAs are the RNAs that are used in making up ribosomes, right? Another group of RNAs, these are called the transfer RNAs, right, or tRNAs. They form the adapters that select amino acids and hold them in place on a ribosome for their incorporation into proteins, right? How many of you are from the communications area? So you know about coding and decoding, right? You will need some kind of a decoder, right? Because three of those letters in the RNA, they will spell out one amino acid, right? But you have to make sure that the correct amino acid is picked up, right, and put into the string to produce the appropriate protein, right? And we will discuss the details of how that is done, right? So the transfer RNAs, they are like the decoders or the adapters that will make sure that the correct amino acid is put at the correct location, right? Now, in the case of eukaryotes, again, what are eukaryotes? What? Yeah, it has a nucleus and other organelles, okay. And the opposite of that are prokaryotes, right? Those are like bacteria that don't have any of those organelles inside, right? So in eukaryotes, each gene coding for a single protein is typically transcribed into a separate mRNA molecule, right? So for every protein, you have a separate mRNA molecule, right? In bacteria, a single mRNA is often transcribed from several adjacent genes, right? And therefore, it can contain information for several different proteins, right? In fact, they are transcribed as what are called operons, right? Several different genes, right? That will transcribe for several different proteins, but usually those proteins, they act together to perform some function. Now, the way in which transcription starts, right, or is initiated, it also differs somewhat between prokaryotes and eukaryotes, right? Between bacteria and, let's say, human cells, it's different. So let's look at these differences. So in the case of prokaryotes, that is like bacteria, the RNA polymerase molecules will slide rapidly along the DNA, right? And the molecule will tightly latch onto the DNA when it encounters a region which is called a promoter, okay? which contains a sequence of nucleotides indicating the starting point of RNA synthesis. So in order for RNA synthesis to take place, you need a specific sequence that is indicating to the RNA polymerase that, hey, this is the place where transcription needs to start, right? So once the RNA polymerase detects that, it'll open up the double helix immediately in front of it, right, and expose nucleotides on a short stretch of DNA on each strand. Again, I mean, as I'm going through this, if you have any questions, ask me, stop me, all right? Now, one of the two exposed DNA strands will act as a template for the synthesis of the complementary RNA. Okay. And then the RNA chain elongation continues until the RNA polymerase encounters a second signal in the DNA, which is the terminator or the stop site. Right? And here the polymerase is going to halt. It is going to stop transcription and release both the DNA template so it can form the double helix and the newly made single-stranded RNA chain. Right? That's how transcription happens in the case of prokaryotes or bacteria. Now, a subunit of bacterial polymerase, which is called a sigma factor, is primarily responsible for recognizing the promoter sequence of DNA. And in fact, here I can show you the picture, right? I think a picture is worth 1,000 words, and here maybe more than that. All right, so here at the top, you have the double-stranded DNA. The top strand is from 5' to 3', left to right, okay? The bottom strand is going from 5' to 3', right to left, right? And here the template strand is going to be the bottom one, right? So this is the RNA polymerase, this blue thing here, that is the RNA polymerase. And then basically there is going to be a promoter region, right? See, you can see the promoter here, right? So the RNA polymerase is going to latch onto the DNA, it's sliding along this until it reaches the promoter region, right? Now, there is something called the sigma factor, right? This is a subunit of this bacterial RNA polymerase. This is the thing that is going to help, right? Like if we go back, see, a subunit of bacterial polymerase that is called the sigma factor, that is primarily responsible for recognizing the promoter sequence on the DNA, right? So you have bacterial polymerase with the sigma factor embedded in it, the yellow one, yellow ball, all right? So it figures out that this is the promoter, okay? Once it reaches the promoter region, then it will start the transcription, right? This blue stuff here, this small strand here, that is the RNA molecule that is being transcribed, right? Looking at the bottom one as a template, right? So you can see the bottom one, 5' to 3' is right to left, right? So if that is used as a template, then for this RNA strand, it's going to be, again, transcription going from 5' to 3' as this thing moves along. Okay? And then this guy, this sigma factor, it is needed only for detecting the promoter region. So after like about 10 nucleotides are synthesized, right? 10 ribonucleotides have been synthesized, then this sigma factor just drops off, right? But this thing continues because the procedure has already, the transcription procedure has already been started. Then this continues all the way until it reaches the stop, right? This red is the stop, right? When it reaches the stop here, then it is going to leave the, it is first of all going to release this RNA, right? From the DNA strand. The double helix will again form, right? And then this guy is also gone. The RNA polymerase is gone. And before it can transcribe again, it will need to bind a sigma factor, right? And then again go along the DNA looking for the promoter region, right? So that's how transcription is. Transcription takes place in the case of bacteria, right, or prokaryotes. So this is the picture. So I've tried to explain everything using this picture, right? Any questions? Otherwise, yeah, go ahead. Does the what? I don't know whether, I don't think it knows it a priori. When it gets there, it'll stop, looking at the sequence. Yeah, this is the stop site over here. Yeah, I don't think it knows it a priori, because it'll stop when it gets to the stop site. So as I pointed out in the picture, a subunit of bacterial polymerase, which is called the sigma factor, is primarily responsible for recognizing the promoter sequence on DNA, right? And once the RNA polymerase has latched onto the promoter and it has synthesized about 10 nucleotides of RNA, the sigma factor is released, right? That's what I showed you in the diagram. And the RNA polymerase moves forward and it continues the transcription process, right? After the polymerase reaches the terminator sequence and it is released, polymerase is released, it reassociates with a free sigma factor and is ready to start transcription again from a promoter region. Maybe for the same gene or for some other gene, okay, whichever it needs to be transcribed. Right? And the figure that I showed you is a schematic diagram showing an RNA polymerase molecule transcribing a bacterial gene. Right? And here the lower DNA strand serves as the template for RNA synthesis, and that is actually clearly indicated in the figure. Right? This is the lower one is the template strand. Okay, now transcription, just like DNA replication, transcription can also proceed only in the 5' to 3' direction, right? Go look back at your figure, okay? See 5', and then this is the 3' end, 5' to 3'. Okay. Moreover, a portion of DNA can be transcribed only if it is preceded by a promoter sequence, right? If you do not have a promoter sequence, that portion cannot be transcribed, right? And this ensures that unnecessary transcription does not take place, you know. A cell should be optimizing its energy, its resources. It shouldn't, go around doing unnecessary transcription. Right? Now, since prokaryotes, right, like bacteria, they don't have a nucleus, so prokaryotic transcription will take place in the cytoplasm, right? Because there is no nucleus or other organelles, right? But in the case of eukaryotes, it's different because eukaryotes, they have the nucleus. The DNA is contained inside the nucleus, right? Most of the DNA. So transcription will take place inside the nucleus, right? And translation takes place outside in the cytoplasm. Right? Now, so if you're looking at bacterial transcription, as mRNA molecules are transcribed, ribosomes will immediately get to the 5' end of the RNA transcript and it'll start protein synthesis, even before the transcription is over. Right? Even before this thing has been released, okay, maybe transcription might be going on, the ribosome could get on here and start doing the translation. Right? And we'll see how it does the translation, right, in a few minutes. All right, any questions? Yeah, go ahead.
So you're saying that the transcription takes place from 5' to 3'. So does this mean that only the sugar is involved? The sugar we have, like deoxyribose or ribose. Are we talking about the carbons then?
No. You're talking about when I say 5' to 3'. Yeah, the 5' to 3' is determined by the, by basically the sugar in there, okay? But here more than the sugar is involved because remember I showed you one picture when I did the structure of DNA, right? One schematic diagram, you had a dot on one side, yellow dot on one side and something else on the other. So although the polarity came from the sugar, but each nucleotide now has one 5' end with that dot, okay, with that phosphate group, and the other side it is a 3' end because it's the hydroxyl group on the sugar, right? But it is a full-fledged nucleotide, like it has a nitrogenous base, it has a sugar, and it has a phosphate, right? Yeah. So yeah, the 5' and 3' terminology are coming from the sugar, right? But here you have more than the sugar, right? You do have a nucleotide with a base, with a sugar, and with a phosphate. Yeah. Did that answer your question?
Yes.
Any other questions? Now, in the case of eukaryotes, right, like say for example in the case of human cells, right, the DNA is enclosed within the nucleus, right? So the transcription, that is the copying of the DNA into RNA, that takes place inside the nucleus. But the protein synthesis takes place out in the cytoplasm. So that means you have to find a way of getting this transcribed RNA out into the cytoplasm, right, before protein synthesis can actually take place, right? So before a eukaryotic mRNA can be translated into protein, it must be transported out of the nucleus and into the cytoplasm, right? And before that happens, some processing takes place, right? So before the RNA is exported from the nucleus, it goes through several different RNA processing steps, right? And depending on which type of RNA is being produced, these transcripts are going to be processed in different ways before leaving the nucleus, right? Now in eukaryotes, the RNA that is produced by transcription but not yet processed, right, for being sent out is often called the primary transcript. So you have the primary transcript, then you have to process it before you get the actual transcript that goes out. Now primary transcripts that are destined to become mRNA molecules. Right? Remember, mRNA molecules, the messenger RNA molecules, these are the ones that will get translated into protein. Right? So for these guys, there are two processing steps that are involved. The first one is the addition of a methyl guanine cap, right, or methyl G cap. So we already know what guanine is. I discussed the structure of that. I'm not asking you to memorize, but at some location on that guanine, if you put a methyl group, again, what is the methyl group? CH3, yeah. So if you put a methyl group, then you get methyl guanine, right? So there will be a methyl guanine cap at the 5' end of the messenger RNA, right? So that gets added after the RNA polymerase has synthesized the 5' end of the primary transcript and before it has completed the transcribing the entire gene, right? So at one end you have the 5' methyl G cap, and at the other end, the 3' end of the eukaryotic RNAs, they are trimmed at a particular sequence of nucleotides, and then a whole bunch of A's, right? Adenine nucleotides are added at the cut end, right? And this second step here is referred to as polyadenylation, right? So you are adding a lot of A's at the 3' end, okay? So if you're looking at eukaryotic messenger RNAs, they will be characterized by the presence of a 5' methyl G cap, all right, and a poly A tail, right? Many adenines, right? Maybe 100, 200 nucleotides are added at the 3' end, right? So that's how you can tell that it's a messenger RNA, right, versus ribosomal RNA, tRNA, and so on, okay, because of this pre-processing. So the trimming and nucleotide addition at the 3' end are carried out by two different enzymes, and the sequence of adenine nucleotides is referred to as a poly A tail, right? Just like you have a methyl G cap, right? 5' methyl G cap, so you have a 3' poly A tail, and it's generally a few hundred nucleotides long. And these two modifications, right, putting a cap, 5' methyl guanine cap, right, and a 3' poly A tail. So these two modifications, capping and polyadenylation, are thought to increase the stability of the messenger RNA molecule and to aid its export from the nucleus to the cytoplasm. Right? And they also later serve to indicate to the protein synthesis machinery that both ends of the message are present and therefore the message is complete. Okay? So the cells have figured out how to do, like in communication you do all this parity checking and all the error correcting codes, and the cells have done it, okay, long before us, right? So these are already done, okay. So that if the methyl G cap and the poly A tail are intact, right, then you know that probably the message has not been tampered with, that you have the correct message. Now, unlike bacterial genes, right, eukaryotic genes contain both coding and non-coding regions. Remember I said earlier that if you're looking at the human genome, you have like 3 billion of those letters, but only about 2% of them, right, code for anything, okay? So the rest of it is non-coding, right? So if you're looking at eukaryotic genes, they contain both coding and non-coding regions. And the coding regions, they are called exons, right? And they are interrupted by non-coding regions that are called introns, right? And the exons are usually much shorter than the introns simply because, I mean, only about 2% is coding, right? And the coding portion of a gene is often only a small fraction of the total length of the gene, right? For RNAs that are destined to become, for eukaryotic RNAs that are destined to become messenger RNAs, right, the introns are removed after capping and polyadenylation, right? And this is carried out in the nucleus before the mRNA is sent out to the cytoplasm, right? And the process is known as RNA splicing, and it will result in a functional mRNA molecule with no non-coding RNAs in between, right? Yeah.
So we are saying that the genes are containing coding and non-coding regions.
Right.
So the genes themselves, one gene gets converted into a protein.
Yeah, see, what is going to happen is that the gene has got both coding and non-coding region. Both the coding and the non-coding region will get transcribed into RNA, okay? They'll get transcribed. However, before the translation takes place, the non-coding portion will be thrown out, okay? That's how it works, yeah. Non-coding portion will be thrown out and you will have a continuous coding sequence, right? And that's the thing that will get translated.
So if there are some mistakes in the non-coding region, that won't affect.
That will not affect the final protein. Yes, that's right. And
So
yeah, About 3 mistakes every time you're copying, because it's one mistake in a billion. Yeah.
So
Yeah.
3 mistakes, and so the probability of that mistake lying in a gene itself would be like less.
Yeah.
And further, like since most of it is the non-coding part, lying in the coding part only then will it create a
Yeah.
problem.
Yeah. Yeah. No, that's the reason why we are not, all of us are not getting cancer every day. Okay. Because, there are lots of checks and balances, so yeah. Yeah, that probability is pretty low, right? But if it does happen in the coding region, so in cancer what has happened is that in the coding region there is a mistake, okay? Because if you keep on doing it, the probability might be low, okay? But once the event occurs, then you're in trouble, okay? So that's what is going to happen. Yeah, if it happens in actually see in the non-coding region also there could be some problems because we don't understand all of that stuff. But even in the non-coding regions, there could be things that are called microRNAs and all that, right? That are coming because they might interfere with something. Like it may not get translated into protein, all right? But on the other hand, suppose some RNA got, some DNA got transcribed into RNA, all right? Then it is going to get translated into protein, all right? Now, maybe to prevent that, the cell might produce a complementary RNA, right, from somewhere, like a microRNA, and make it like double-stranded for a portion, okay? So that doesn't get translated. But if that thing got messed up, all right, the thing for the microRNA, then that protein might get produced. So, the picture is not that clear, right? And it's up to you guys to come and clarify that, okay? Because you do need people with strong quantitative skills to work on that. The biologists by themselves will not be able to clarify that because it's 3 billion letters, okay? I mean, and when you say the human genome has been sequenced, means you just found out what all those letters are, right? You don't know what the function is, okay? What parts of those, and how everything is linked, okay? Which gene is affecting which one, okay? Those are some of the things that we are interested in unraveling. It's not easy because the kind of data that you get usually doesn't have causality, right? This is not a man-made engineering system, right? So you're trying to do reverse engineering based on data, okay? Data doesn't have causality. Yeah, there is correlation, right? But how do you get causality from that? That's a different issue. Yeah, so it's not easy. Like, and that's why many times you find in cancer somebody figured out that this is the gene that's causing that cancer. Okay, you go and knock that gene out. Guess what? The cancer comes back with full force. Okay, why? Because maybe you knocked out one of the drivers. All right, but there's a lot of redundancy and crosstalk. Okay, some other genes are affecting that. All right. So how do you knock that out? Right? You don't because if you don't know your enemy, right? How are you going to attack the enemy? Right. So it is a problem, and I think that it's going to take a long time, but I think you need people with different kinds of skills to come together, right, and work on it because this is different. Like even the most complicated man-made system, we built it, okay, whether it's communication network or power system or whatever. So we know. I mean, we may not like to build such a detailed model, but the grid and we can do those things, right? It can be done. But here is some system that we have not built, okay? We are trying to unravel it, right? We don't even know what affects what, and it's a very complicated multi-input, multi-output system, right? So with links that are not fully known, and you want to basically get some good behavior out of it. Right? That's where we are headed because all this stuff I'm covering to just impress upon you the complexity, and you need to have domain knowledge. Okay, you cannot come and say I know Viterbi's algorithm. Show me a problem in biology where I can apply Viterbi's algorithm. No, you need to understand the biology so that even if you apply Viterbi's algorithm to something, it makes biological sense. And it's not some artificial application that you've done. It's like, for example, if you have a hammer, okay, everything to you in the world looks like a nail, all right? You don't want that. So you want to forget about the hammer. You have the hammer in your head. You just keep it with you, all right? Go and study the other problem, all right? And then see, okay, if in your toolbox you have something that will solve that problem. If not, you try to devise, and that's the way research should evolve, all right? So in this course, I mean, this is something I'm trying for the first time. That's why I'm going a little bit fast. If it's a problem for you, let me know, okay? But based on asking you guys questions, it looks like you're keeping up pace, right? I'm going a little bit faster than I have in the earlier semesters. Why? Because I want you to not only for cancer and for, animal biology, right? But I also want to give you the ropes for doing plant genomics, okay? And by the way, there is a seminar tomorrow. I don't know whether you know about it or not. From 4:10 to 5, I think it is in chemical engineering building 108, right? there is somebody from AgriLife who will come and talk about the use of UAVs, all right? Unmanned autonomous vehicles, all right? Well, in everyday language, we refer to them as drones, although that's not politically correct. Drones are for knocking out your enemies and all that. But they're using UAVs, all right, to basically take pictures, all right, of, let's say, cornfields or maize fields from the air, right? So you're going to take pictures and you're going to use hyperspectral imaging, right, to figure out, let's say, what the nutritional status is of those plants, okay, and then apply fertilizer and or water and all that as needed, okay. So this is a pretty exciting project that involves a lot of faculty, right? It's a very big team, and they published a paper recently in PLOS ONE, okay. I can give you the link for that, right, for those of you that might be interested. They published that paper in PLOS ONE. There are a lot of people, I think maybe there are 50 people involved, okay, across the campus. But this is a high priority project both for the College of Agriculture and for engineering, right? So, you know, that might be and I think there are, like, some of you probably know Professor Chamberlain, right? I mean, he's involved in this project already, and we're trying to get more of our faculty involved, right? Because, in fact, you know, at the center, we have somebody that is helping them with image processing and all that stuff, you know. So, you know, there might be lots of different things but you need this background, the background that I'm covering, because otherwise it's like, you know, you're in a foreign land, you don't know the language, and you're trying to do something, you know. And what you do may not be a breakthrough in terms of EE contributions, okay? But if it makes a big difference, right, in such a big project, right, you can imagine that the payoff could be huge, right? So I can send you the information. I think it's in room number 108 of the Chemical Jackie Brown building, and I think it's at either at 4 o'clock or 4:10 tomorrow afternoon. I'll try to send you the link, okay, after the class. So you might want to go there. That's tomorrow. That's Friday. I will be there, and I've asked a lot of other people also to go there, you know. So, all right. Any other questions? So this process of removing the non-coding region, that is referred to as RNA splicing, right? And it will result in a functional mRNA molecule with uninterrupted coding sequences, right? So a natural question that comes up is, how does the cell determine which parts of the primary transcript should be taken out, right? And it turns out that each intron contains a few short nucleotide sequences that act as cues for its removal. And these sequences are found near each end of the intron and are the same or very similar in all introns. And introns are removed from RNA by enzymes that are composed of a complex of protein and RNA. So these enzymes are not just protein, right? Protein and RNA together, okay? And these splicing enzymes are called SNRPs, or small nuclear ribonucleoprotein particles, right? And let me show you the picture here, okay? How it's removed. See, this is exon A. This is 5' end, 3' end, right? This is like the primary transcript before it has been spliced, exon A and exon B. This is the intron in between, right? So the two things will come together. SNRP will bring the two ends together, right? And then it'll cut this guy out, right? And produce a single mRNA molecule, right? Exon A, exon B. Not only this, okay, you can have, so this procedure is called splicing, but you can also have what is called alternative splicing. Let's say you had exon A, you had an intron, exon B, right? Another intron and an exon C, right? Then there are different possibilities. You could cut out the intron the exon in the middle completely, and you could say A and C together are forming the mRNA molecule, right? Or it could be A, B, C, right? So there are many possibilities. So starting from the same primary transcript, the cell has the ability to produce different messenger RNA molecules. This is referred to as alternative splicing. It can make a big difference because the protein that you ultimately get is going to be completely different, right? Now, to get back to your point, the presence of numerous introns in eukaryotic DNA, So the presence of numerous introns in eukaryotic DNA makes genetic recombination between exons of different genes more likely. Just like you said that, I mean, because as long as the coding region is not messed up, right, things will still be okay, right? That means if you have, if you have some, two very similar sequences of DNA, they line up next to each other, and there is a crossover. We'll see how that takes place later. But if there is a crossover, now if the DNA had only coding region, right, from the crossover you might get loss of function, right? because the coding region might get messed up. But the fact that you have so many introns means that portion can get messed up and still you can have a functional gene, right? And this will be discussed in detail in a later chapter when we talk about genetic variation, right? So here we note that eukaryotic genes for new proteins could have evolved quite rapidly by the combinations of parts of pre-existing eukaryotic genes, because the introns, even if they get messed up, nothing is going to happen, right? So you can still have a functional gene. Moreover, the primary transcripts of many eukaryotic genes contain 3 or more exons, and this is what I was trying to show you. Like if you have ABC, what is going to happen, all right? Can be spliced in various ways to produce different messenger RNAs, right? From the same original coding region, right? From the same coding DNA, depending on the cell type, right? Some cell might need the protein that you get from ABC. Some other cell might need BC, right? Some other cell might need AB. Some other cell might need just AC, right? So by alternative splicing, it can produce different proteins in different cell types, right? And this is referred to, so this is the phenomenon of alternative splicing. Again, this is just terminology. And again, if you talk to a biologist when they talk about alternative splicing, it's like ABC to them, you know. It's like, you know, like Ohm's law to us, okay, something like that, okay? Ohm's law, KCl, KVL, and all that stuff. So it's important to know the terminology. So at each intron, a group of these snRNPs, they will get on this, and they will cut this fellow out, and they'll rejoin the RNA chain, and releasing the excised intron as something that is called a lariat, right? And that's going to be recycled inside the cell, right? But you will now get this messenger RNA molecule. Now the length of time that an mRNA molecule persists in the cell affects the amount of protein produced from it. And different mRNA molecules have got different lifetimes, right? I mean, there might be some that might last for seconds, some for hours, you know, or days or whatever. So they're different. Now, although the genomes of current day prokaryotes do not contain any introns, bacteria do not have any introns, right? It's all, everything is exon, okay? Everything is coding region. It is thought that their earlier ancestors may have had introns in their genes, and they might have evolved and gotten rid of their introns in order to have a smaller genome, right? Genome again is the set of all the letters that you have in the DNA of that organism. So so over evolutionary time they got a smaller genome which would allow faster DNA replication because they have to copy less number of units, right? Thus facilitating very rapid reproduction, which is a characteristic that current day prokaryotes do possess. Like if you look at E. coli, it'll reproduce every 20 minutes. That means in 20 minutes it has to copy the entire genome, right, and make a copy of itself, right, new copy of itself, right. So if you don't have non-coding regions, all right, you can do it a lot faster, right. So probably over evolutionary time, this is what prokaryotes did to themselves. Yeah, go ahead.
Is there a trade-off between like the accuracy with which we are replicating and the rate at which they are reproducing?
Yeah, there is a trade-off, yeah. If you reproduce fast, you'll make more mistakes, okay? Like if you're looking at a human cell, okay, in order for DNA mutations to take place, you have to wait a long time, right? But bacteria, like in one day, it'll go from 1 bacterium to 1 billion, right? So you can see mutations accumulating a lot faster because they replicate faster, right? In fact, if you treat a bacteria with antibiotics, right, it's supposed to kill the bacteria. So let's say you have 5 billion bacterial cells, okay? Most of them are dead. Maybe 1 or 2 survive, which are resistant, that are mutant. That will take over the population, right? So in other words, it's watching a movie in fast forward, right? What would happen to a human being may take thousands of years, right, or millions of years. You can see in bacteria that will happen in real time. In one day you will see that happening, right? Because every 20 minutes it's reproducing, right? Okay, that concludes our description of transcription, right? And now we are going to move on to translation. There is more to transcription in, see, like in bacteria it's kind of simpler, okay, like how transcription takes place, but if you're looking at eukaryotic cells, all right, the control of transcription is much more complicated because in the case of in bacteria it might be just one factor is coming in and binding and transcription takes place, right? In the case of eukaryotic cells, it's like a whole committee is deciding whether transcription will take place or not. And in fact, the next chapter, right, not this one, next chapter is actually devoted to that, where you talk about chromosomes and gene regulation. What decides when the genes are going to turn on and get transcribed? So it's a lot more complicated in the case of eukaryotic cells, right? But now so that's an entirely different chapter in its own right? Now let's talk about translation and try to understand, the basic mechanisms of how translation is brought about. So in translation, the sequence of nucleotides in a messenger RNA molecule have to be decoded to produce the appropriate proteins, right? And the code that is used by nature for this purpose is called the genetic code, right? And it is the map from the nucleotide-based language of RNA, right? Because DNA has been copied into RNA, and then that is going to be used to figure out what protein to produce, right? So it's the map from the nucleotide-based language of RNA to the amino acid-based language of protein, right? And the genetic code was completely worked out in the early 1960s, right? And at least, people that worked it out, right? There may be several, but at least I know one person that did get a Nobel Prize for that, and many of you may have heard of Hargobind Khurana, right? His Nobel Prize is basically related to this translation thing, okay? And people had to, see, first of all, people had to spend a lot of time to figure out that it is actually a triplet code, right? Because remember, if you're looking at RNA, in every location, how many possibilities are there? How many? 4 possibilities, okay? A, G, C, U, okay? So that means if you have a 1-letter code, you can only code for 4 amino acids. If you have a 2-letter code, you can code for how many? 16, okay? That's still not enough because we know there are 20 amino acids. So at a minimum, you need a 3-letter code, right? Right? But it could have been more. It could be 4, it could be 5, right? So people had to do experiments because if it's a 3-letter code, then if you shift it by 3 units, right, you will get exactly the same amino acids back, okay? So people have done, so I mean, a lot of people have got Nobel Prizes for these, but these are, groundbreaking discoveries that people made, right? So is it really triplet code, right? A lot of experiments have gone in. So we are just, presenting like the final result, right? Just like in electrical engineering, if it's Fourier series, Fourier transform, you don't look at, how much time Fourier spent on all these series. You just get the final result and even the Laplace transform. So it's the same thing here, right? We're not worrying about that, but I'm just telling you that a lot of effort did go in, right, to figure out that this is actually a triplet code. So it's confirmed by experiments in the 1960s. And the sequence of nucleotides in the messenger RNA molecule is read consecutively in groups of 3, each such group specifying either the start of a protein, the end of a protein, or an amino acid, right? So there is and these are called codons, right? Each nucleotide triplet is called a codon. So you have a start codon, you have a stop codon, right? So you have you have 3 of them, right? I mean, so in each location there are 4 possibilities. That means you have how many codons altogether? Huh? No. How many codons are there possible altogether? Right? 64 codons. Yeah. Don't be afraid. Okay. This is like quantitative stuff. This is your home turf, right? I mean, in biology we cannot make it a different number. So there are 64 possible codons, right? So out of that, there are 3 of them are stop codons, right? So that means there are 61 that are remaining, right? But you have 20 amino acids, right? That means there has to be more than 1 codon, okay, that codes for some amino acids, right? So it's like a degenerate code. So we are getting to all that. So there are 20 possible amino acids and there are 64 possible codons. These codons, UAA, UAG, UGA, these are stop codons. And they will signify the end of translation, right? Now the codon AUG, right, it specifies 1 amino acid which is called methionine. Remember we talked about 20 amino acids, right? You're not responsible for remembering all the names. Methionine is one of them. And also it indicates the start of a protein. So whenever you're doing translation, it will always start with methionine. That methionine might be removed later on, okay? Because that codon OG codes for methionine, right? All the other codons, so other than the stop codon, right, the 3 stop codons and then the codon OG, AUG, which codes for methionine, all the other codons, they code for some amino acid, right? Any questions so far?
What happens if OG is there in between? I mean, how
No, if OG is there in between, that will be methionine, okay? That is going to be methionine. The OG in front, that is going to be taken out. There are detailed mechanisms, So we'll probably be shedding some light as we go along, right? But you know, to understand the details, you probably have to read more. Okay, more than at least the level at which I have read it. Okay, but I think from the explanation I give you about how the translation takes place, you will see that you know the org is the tRNA in the case of org is bound to a special location. You know, so it's taken out at the end. You know, so I think your question will get answered, but again, I mean, like you may have to read more. Right? So the sequence of nucleotides in the messenger RNA molecule is read consecutively in groups of 3, each such group specifying either the start of a protein, that's like the OG, right? The end of a protein would be one of those 3 codons that are starting with U, right? Or an amino acid. And each nucleotide triplet is called a codon, right? So there are 20 possible amino acids and there are 64 possible codons, right? Yeah, actually I'm repeating this slide. I thought I was on the next page. So since there are 20 different amino acids and 61 codons coding for amino acids, there must be multiple codons coding for the same amino acid. Okay? In other words, the genetic code is what is called degenerate, right? The genetic code is also universal, right? In the sense that all known life on this planet makes use of the same genetic code. Now, if you find life on Mars or some other planet and there is a genetic code used, it might be different, right? We don't know, right? But on this planet, all living organisms use the same genetic code. And we have not again included the entire genetic code here since it is not crucial for our purposes. But if you open any book on molecular biology, they'll have the entire genetic code. Okay, for all the 64 codons, you know, like which amino acid they map into, you'll get methionine, you know, histidine, tyrosine, and so on. Okay. And if you, like probably if you're like aspiring to become an MD or a doctor, or but you probably memorize all those things, all right? I mean, I'm not asking you to memorize all those different amino acids and then you know the codons that code for them, but if you have an interest in that and you have very strong memory, just go ahead. You know, I'm not holding you responsible to that. Now, since each triplet of nucleotides codes for an amino acid, an RNA sequence can be translated in any one of 3 reading frames. Right? By shifting it by 1, right, it'll be a different reading frame, depending on where the decoding process begins. Right? So the choice of the reading frame is important, since advancing the reading frame by even 1 nucleotide, right, can alter all the amino acids. Right? Like if you had AUG, you just left out the A. Okay, now you have UG something, so it's a completely different amino acid, most probably. All right. And so it'll have a completely different 3-dimensional conformation for the protein, right? And it'll cause loss of function, right? So we'll see later how a punctuation signal at the beginning of each message will set the correct reading frame. Okay, we'll look at that later on. Now the triplets in an mRNA molecule specify the sequence of amino acids that have to be linked together to produce the particular protein. And to actually produce that protein, you need some kind of a decoder or adapter molecule is needed. And those decoders are called transfer RNAs, tRNAs or transfer RNAs. They are RNA molecules, right? So tRNAs serve as adapter molecules that can recognize and bind both to the codon on the messenger RNA and at another site on their surface to the corresponding amino acid, right? Because you have to make sure that the right amino acid is getting incorporated in the protein. Now, a tRNA molecule consists of a chain of about 80 ribonucleotides. It's an RNA, right? So it has to have ribonucleotides, about 80 of them. And this is what it looks like, right? A typical tRNA molecule, right? So you have a loop that is called a D loop, then a T loop. So here there is base pairing that is taking place on the same molecule, hydrogen bonding, right? Between A and U, G and C, right? So it is assuming this shape. The important thing, so this is the 5' end of the tRNA molecule. This is the 3' end. Right? The important thing to note is that over here there is what is called an anticodon, right? This is going to pair, so there are 3 nucleotides here, right? This anticodon is going to pair with the codon, right? So like if you have UAA, all right, then on this anticodon, right, this is going to be AUU, right, the complementary one. And so it will have some sequence here. Each of these tRNAs will have some sequence here that pairs with some codon, right? And then it will have something, so it will have some mechanism by which the correct amino acid is going to get bonded here at the 3' location. So then you will have a charged tRNA, right? So you have the correct code attached to the correct amino acid, right? And this thing plays an important role in carrying out the synthesis of the protein. Okay, and I will explain to you in details how that is done. And that's a question that you can expect on your exam. Okay, how is this thing done? Okay, explain the steps. Any questions so far? So for the tRNA that we just showed in that diagram, parts of this chain assume particular shapes by complementary base pairing, as we showed already. However, two regions of unpaired nucleotides are crucial to the function of tRNA in protein synthesis. First one is the anticodon, which is a set of 3 consecutive nucleotides that pairs with the complementary codon in an mRNA molecule, in a messenger RNA molecule. Right? And the second one is a short single-stranded region at the 3' end of the molecule, which is the site where the amino acid that matches the codon is attached to the tRNA. All right, any questions? Now, because the genetic code is degenerate, it implies that either there is more than one tRNA for many of the amino acids, right? Or that some tRNA molecules can base pair with more than one codon, right? Because you have 20 amino acids, right? And you have like 61 possible tRNAs, right? I mean, those anticodons, 61 possible anticodons, right? So in fact, both these situations actually occur. Some amino acids have more than one tRNA, right? That means with different anticodons, right? With the same amino acid attached, right? And there are some tRNAs which are constructed so that they require accurate base pairing only at the first 2 positions, right? And of the codon, and they can tolerate a mismatch or wobble at the 3rd position. So the 3rd position is kind of flexible, right? And this is the reason the wobble base pairing explains why many of the alternative codons for an amino acid differ only in their 3rd nucleotide. And an example of that is that the codon CCA, CCC, CCG, and CCU, all of them code for one of the amino acids which is called proline. Okay, so as long as the first 2 locations match, right, you will have the correct protein, correct amino acid. Now, specific enzymes which are called amino acid tRNA synthetases, they covalently couple each amino acid to its appropriate set of tRNA molecules, right? Because at the top, see, the tRNA has got to be attached to its right amino acid through a high energy bond. Why do you need the high energy bond? Because when you are going to synthesize a protein, right, that is an energetically unfavorable reaction because it's an order generating reaction. So you have to break down some high energy bond, right, to make the reaction feasible, because the total free energy change has got to be negative. Yeah. So that's why that's why these tRNAs, these are charged up with these high energy bonds, okay, which are going to be broken to produce the protein chain from these amino acids. Right. So specific nucleotides in both the codon, in both the anticodon and the amino acid accepting arm allow the correct tRNA to be recognized by the synthetase enzyme. Okay, so you have to make sure that the correct amino acid is paired to the correct tRNA. Right? So the synthetase catalyzed reaction that attaches the amino acid to the 3' end of the tRNA is powered by ATP hydrolysis. Again, I mean, you need energy. Okay? You're going to create a high energy bond over there, right? And it produces this high energy bond between the tRNA and the amino acid. And later on, the energy of this bond is used in protein synthesis to covalently link the amino acid to the growing polypeptide chain, right? Because each amino acid is going to be linked to the next one through a what kind of bond? Amino acids are linked together by what kind of bond? Peptide bond. You know? So you're going to have a polypeptide chain with many peptides in there. All right, now, so how does this, so now we have the adapter molecule, right? We've looked at the anticodon. We've talked about how the correct amino acid is going to get attached to the tRNA. So the question is, how is the protein actually going to be produced? Okay. Now the protein is actually produced on molecular machines that are called ribosomes. So the RNA message is decoded on what are called ribosomes. These are large molecular machines that facilitate accurate and rapid translation of mRNA into protein. And the ribosome will travel along the mRNA chain. Okay, remember every 3 units over there, triplet is coding for an amino acid, right? So the ribosome will travel and it will start, producing a polypeptide chain with the corresponding amino acids. So the ribosome travels along the mRNA chain, capturing complementary tRNA molecules, holding them in position and bonding together the amino acids that they carry so as to form a protein chain. So what is the structure of a ribosome? A ribosome is made up from more than 50 different proteins, which are called the ribosomal proteins, and several RNA molecules called ribosomal RNAs. Remember, we said there were three types of RNAs: messenger RNA, all right, transfer RNA, all right, I already explained that, and ribosomal RNA, all right, rRNAs, all right. So a typical living cell contains millions of ribosomes in its cytoplasm, right? And in a eukaryote, right, remember, transcription is taking place inside the nucleus, right? Translation will take place in the cytoplasm. So in a eukaryote, the ribosomal subunits are made in the nucleus by the association of newly transcribed ribosomal RNAs with ribosomal proteins, which have been transported into the nucleus after their synthesis into the cytoplasm. Because for ribosomal proteins, again, I mean, initially the ribosomal RNA is produced inside the nucleus, but then it goes out, gets translated, ribosomal protein comes from there, and then more ribosomal RNAs are there, whatever is needed, everything is put together to produce the ribosome. Right? So the individual ribosomal units are then exported to the cytoplasm to take part in protein synthesis. A ribosome is made up of two units. It has a small subunit and a large subunit, which fit together to form a complete ribosome with a mass of several million Daltons. So it's very heavy, it's a big molecular machine. And the structure of a typical ribosome is shown in the figure on the next page. Right? So this is a ribosome. This one at the bottom is the small subunit. The one at the top is the large subunit, large ribosomal subunit. Now the bottom one, it'll have an mRNA binding site here, right? And this guy has got three other sites: A site, P site, E site. E is like for exit. And I'll show you how the whole thing actually works, right? Now the small subunit matches the transfer RNAs to the codons of the messenger RNA, while the large subunit actually catalyzes the formation of the peptide bonds that link the amino acids together into a polypeptide chain. And I'll explain exactly how that happens with a picture. And the two subunits will come together on an mRNA molecule, usually near its beginning, the 5' end, to begin the synthesis of a protein. So as I showed you, a ribosome contains 4 binding sites for RNA. Okay, so there are 4 binding sites. Okay, one is for mRNA on the small subunit, and then A, P, and E. Right, those are for transfer RNAs. A, P, and E are binding sites for transfer RNAs. So ribosome contains 4 binding sites for RNA molecules. One is for the mRNA, and 3 called the A site, P site, and the E site are for transfer RNAs. Now a transfer RNA molecule is going to be held tightly at the A or the P site only if its anticodon, right, matches or it pairs correctly, allowing for wobble, right, with the triplet codon that is located over here on the small unit. Right? Like if you have AUG, all right, then the tRNA which can be at that location will have to base pair with that AUG. So UAC, right, is what the anticodon should look like. Right? So a tRNA molecule is held tightly at the A and P sites only if its anticodon forms base pairs, allowing for wobble. Okay, we said in the third location there can be a mismatch, right? That's the wobble, with a complementary codon on the mRNA molecule that is bound to the ribosome, that is bound to the small subunit of the ribosome. The A and P sites are sufficiently close together that their 2 tRNA molecules are forced to form base pairs with adjacent codons on the mRNA molecule. So these guys are close enough. Like, so if you have mRNA, every 3 units of the mRNA is a codon, right? So this A and P are close enough so that these guys, the tRNA that you put in A and the P, they will base pair with the adjacent codons on the mRNA molecule. Okay. So having introduced tRNAs and ribosomes, we are now in a position to take a detailed look at the steps that are involved in protein synthesis. And let us first focus on examining the mechanism by which the ribosome adds a new amino acid to a growing polypeptide chain. Right? So we have to look at 3 things. Okay, how does the protein synthesis start? Right? How do you initiate it? How do you terminate it? Right? And the third thing that we have to look at is how does the chain elongate, right? Let's say if I'm given a chain that is 3 units long, how do I make it 4 units long and so on, okay? So first I'm going to focus on how to elongate the chain. So I'm going to assume that it's not initiation. You already have part of the chain that is already there, right? How do I add the next unit? How does the ribosome do that, okay? And then we'll worry about how to start it and how to finish it, okay? So fair enough, right? So the 3 steps involved are shown in the figure, right? And let's not worry about the text on the left right now, right? Let's look at the picture, right? Again, the picture will make it very easy to explain. So here, this is the small ribosomal unit at the bottom, right? And at the top, you have the large ribosomal unit. This is the ribosome, this is the mRNA binding site. You can see the mRNA is bound here 5' to 3', right? And then this is the A site, the P site, and the E site, okay? So in step 1, you have a situation where part of the polypeptide chain has already been synthesized. You have amino acid number 1, 2, and 3. They are already in there. Okay. Then the third amino acid, right, is still attached to its transfer RNA, right, which is at this location at the P site, right. And now you want to elongate the chain. Okay. How do you elongate? Well, the tRNA that corresponds to the codon here will come and bind. That is tRNA number 4, carrying the appropriate amino acid will come and bind at the A site. Okay, this is step number 1. In the next step, what is going to happen is that the bottom, right, is going to shift by 1 codon to the left, right? It's going to and carrying those tRNAs along with it. As a result, the tRNA for amino acid number 3 will now move to the E site, right? The tRNA for amino acid number 4 will move to the P site, okay? And at the same time, this bond here is broken and this is linked together. Remember, this was a high energy bond, right? High energy bond is broken and 3 and 4 get linked together, right? So that's step number 2. And then in step number 3, this thing is going to go back, right? It is sliding back, but it's not carrying the messenger RNA with it. Just this portion slides back, right? So now you have a different codon that has come to this location, indicated in brown, right? So now the new tRNA can come to this location, all right? And then we are basically back to step 1, right? Only difference is that now the chain is 1 unit longer. Right, you see that? Any questions on this? And so this process we'll keep on repeating, like is shown on the next picture. This repeats and the chain elongates, basically. So let me just read through this. So in the first step, the polypeptide chain containing the amino acids 1, 2, and 3 has just been synthesized, and the tRNA corresponding to amino acid 3 is still attached to the amino acid 3 while being located in the P site of the ribosome. Right? That's what's being shown in that picture. Right? The codon for amino acid 4 being located on the mRNA in the A site of the ribosome. The corresponding tRNA with its bound amino acid comes and occupies the A site of the ribosome. Right? There's this thing here. Right? And in the second step, the small ribosomal subunit with the bound messenger RNA moves relative to the large ribosomal subunit in such a way that the tRNAs 3 and 4 get shifted to the E and P sites respectively on the ribosome. Simultaneously, the amino acid 3 detaches from its tRNA and links up to amino acid 4, still attached to its tRNA. Right? And in the third step, the tRNA for amino acid 3 is expelled from the E site of the ribosome. That's why it's called an exit site. Right? And the small ribosomal subunit moves relative to the large ribosomal subunit so that they line up as before, right? And then the process continues. So you're back to step number 1. Now the central reaction of protein synthesis in which an amino acid bound to its tRNA is detached from the tRNA and linked to the growing polypeptide chain is catalyzed by peptidyl transferase enzyme activity, which is part of the ribosome. Right? Because the ribosome is the machinery that makes this happen. Okay? And it's an energetically unfavorable reaction. So you're breaking a high energy bond. Right? Now the catalytic part of the ribosome in this case is thought to be not one of the proteins, but rather one of the ribosomal RNAs in the large ribosomal subunit. Like somebody asked for an example where an RNA can function as a catalyst, right? Part of the ribosome is the RNA part that actually carries out this catalysis, right? Or at least it's thought to carry out, right? So now what is remaining? So I have shown you how the protein chain is going to elongate, right? The peptide chain is going to elongate. Now the question is, how does it start and how does it end, right? So we'll try to explain that. Before that, any questions on this elongation? Now, codons in the mRNA signal where to start and where to stop protein synthesis. The translation of an mRNA begins with the codon, AUG, right, which also codes for methionine, right, and a special tRNA is required to initiate translation, right. The initiated tRNA always carries the amino acid methionine, and in bacteria, a modified form of methionine, which is formylmethionine, right, is used, right. So that newly made proteins all have methionine as the first amino acid at their amino terminal end. And this methionine, this initiator one, is going to be removed by a specific protease. Again, proteases are enzymes that degrade proteins. Okay, we know that. Right? So the initiator tRNA is distinct from the tRNA that normally carries methionine. So there is a distinction here, like how you were asking, how does it know, right? The initiator tRNA is different from the tRNA that you will have in the middle of the codon. So the initiator tRNA is distinct from the tRNA that normally carries methionine. And in eukaryotes, the initiator tRNA, which is coupled to methionine, is first loaded into the small ribosomal subunit along with additional proteins that are called initiation factors. Of all the charged tRNAs in the cell, only the charged initiator tRNA is capable of tightly binding the small ribosomal subunit as shown in the figure. Let me show you the figure. See, this is the initiator tRNA, right, with methionine attached to it. Right? AUG, it's going to base pair with AUG. It is the only tRNA which is capable of binding the small ribosomal subunit at the P site. Okay? So with this bound, this is going to go searching. It's going to go looking until it finds the first OG, right, AUG. Once it finds the AUG, there are some other factors that will dissociate. They will make room for this big subunit to basically get on this. Okay? Once that happens, now look, all you need is basically a new tRNA to come and bind here, and you're back to step one of what I showed of the chain elongation. Okay, that's how it starts, and then large ribosomal subunit is assembled, and then the first amino acid, the real amino acid, not the initiation one. Okay, the real amino acid, not methionine, will come to this location, and then exactly the same steps as before will take place. This thing will move to the left and then come to the right and so on. Right? So that's how it occurs, and let's just go through the textual part now. So of all the charged tRNAs in the cell, only the charged initiated tRNA is capable of tightly binding the small ribosomal subunit as shown in the figure, and the loaded ribosomal subunit binds to the 5' end of an mRNA molecule, which is recognized in part by the methyl G cap. Again, for eukaryotic mRNAs, there's a methyl G cap, right? So it recognizes that. The small ribosomal subunit will then move forward along the mRNA, searching for the first OG, AUG, right? When this AUG is encountered, several initiation factors will dissociate from the small ribosomal subunit to make room for the large ribosomal subunit to assemble and complete the ribosome. And that's shown in this picture. And once the ribosome assembly is completed, the next tRNA with attached amino acid can bind the codon in the A site, and the protein synthesis can continue as described earlier. How the chain elongates, the same process happens. Now the mechanism for selecting a start codon in bacteria is different because the bacterial mRNA do not have methyl G caps, right? So bacterial mRNA have no methyl G caps to tell the ribosome where to start searching for the start of translation. Instead, they contain specific ribosome binding sequences up to 6 nucleotides long that are located a few nucleotides upstream of the AUGs at which translation is to begin. Right? And unlike a eukaryotic ribosome, a prokaryotic ribosome can readily bind directly to a start codon which lies in the interior of an mRNA because it's not looking for the methyl G cap, okay? It's just looking for that sequence. Right? So as long as the ribosome binding site precedes it by several nucleotides. So consequently, prokaryotic mRNAs are often polycistronic, that is, they encode several different proteins. Right? And in contrast, a eukaryotic mRNA usually carries the information for just a single protein. So we have discussed the start of translation, right? How the chain elongation takes place. So now we have to see how the process stops. The end of the protein coding message is signaled by one of several codons. All right, these are the stop codons, UAA, UAG, or UGA, that are called stop codons. These are not recognized by a tRNA, and they do not specify an amino acid, but they instead signal to the ribosome that, hey, it's time to stop translation, right? And proteins known as release factors, they will bind to any stop codon that reaches the A site on the ribosome, right? It is not going to be tRNAs, it's going to be proteins that are called release factors. And this binding alters the activity of the peptidyl transferase in the ribosome, right? The catalyst in the ribosome, or the enzyme in the ribosome, causing it to catalyze the addition of a water molecule instead of an amino acid to the peptidyl tRNA. And this reaction frees the carboxyl end of the growing polypeptide. And since only this attachment normally holds the growing polypeptide to the ribosome, the completed protein chain is immediately released into the cytoplasm, right? And so that is the entire procedure of protein synthesis, right? Now, even before a ribosome has finished translating an mRNA, because remember, the ribosome will start from the 5' end of the mRNA. Once it has made some progress, maybe additional ribosomes can go and further upstream and start translation. So it can be producing several, protein molecules at the same time, So like if you look upstream, those are less further along. If you look downstream, you have the complete protein. Okay, the others are being produced, right? So you can see those under a microscope. So even before a ribosome has finished translating an mRNA, other ribosomes can get on the mRNA at more upstream locations and initiate translation. Thus, several ribosomes could be simultaneously working on the same mRNA molecule, forming what are called polyribosomes. And this is something that you can see under the microscope, right? No, no, no. They will give several molecules of the same protein. Yeah, because you have the mRNA, right? I mean, that is being translated to produce the protein. But what I'm saying is that the ribosomes don't have to wait until the entire mRNA has been translated into protein, okay? So one guy got in at the 5' location, started generating a protein molecule, okay? That's elongating. But then as soon as this guy moves away from the 5' location, there's room for another ribosome to get and start producing a second protein molecule, So yeah, simultaneously, and the lengths will be different. The one which will be the longest will be the one furthest downstream, right, and so on. And you can actually see that under a microscope. Now, the amount of protein in each cell is regulated by carefully controlled protein breakdown, and proteins vary enormously in their lifespan. Some proteins may last for months or even years. Others last for days, hours, or even seconds, right? And a natural question that arises is how the cell controls these lifetimes. And cells have specialized pathways that enzymatically break proteins down into their constituent amino acids, a process which is called proteolysis. And again, as I said before in the last chapter, right, enzymes that degrade proteins are known collectively as proteases, right? They break down proteins. One function of proteolytic pathways is to rapidly degrade those proteins whose lifetimes must be short, right? Another one is to recognize and eliminate proteins that are damaged or misfolded. Because if something didn't make it properly in the first go, right, tear it apart and then recycle the components and make it again, right? So most proteins that are degraded in the cytosol of eukaryotic cells are broken down by large complexes of proteolytic enzymes that are called proteasomes. These are like our trash compactors, okay? You put everything in, they break it up, right? And the proteins that are imported into proteasomes for degradation usually have been marked out for destruction by the covalent attachment of a small protein called ubiquitin, right? So this is like a garbage tag, right? So you're tagging the garbage that this needs to go into the proteasome and get degraded. So anything that you can think of, the cells have already figured that out, so how to do it, and quite efficiently, with a small size. Now in all of life that we see around us today, DNA, RNA and proteins have got very specialized individual roles, right? DNA is coding the genetic information, RNA is like the intermediate, proteins are functioning as catalysts, right? So it is speculated that life initially originated in an RNA world, right? Everything was done by RNA. Since RNA is capable of both information storage in the sequence of its nucleotides as well as catalytic functions, because it's a single-stranded molecule, it can fold up, right, by this hydrogen bonding, right? So this seems to be a plausible conjecture. And it is thought that the initial RNA formed during the violent conditions present on the ancient Earth. Like people have done experiments where, if you take an atmosphere that is made of methane, like in the ancient Earth there was no oxygen and all that, methane and things like that. And let's say there was lightning, right? So they have seen that these molecules like RNA, DNA, I'm sorry, not DNA, RNA, molecules like RNA can be produced, right? If you have nitrogen, you have, nitrogen, methane, and so on, right? You do some heavy-duty electric arcing, right? They produce those molecules. So that's what people conjecture, We don't know whether that's true or not. So later on, DNA and proteins took over some tasks from the RNA. Since DNA was better suited for information storage because in the sequence of its nucleotides, very stable molecule, and proteins could perform a whole variety of other functions like catalyzing, right? And resulting in the state of affairs that we see around us today, right? So that actually concludes this chapter, right? And let's stop here for today, right? Any questions?
There's one more question about transcription.
Mm-hmm.
We use one DNA string as template, template string. So how should we decide which string is the template?
See, remember RNA polymerase, right, will always transcribe in the 5' to 3' direction, right? So RNA polymerase will see which one it can actually transcribe, yeah, 5' to 3' direction, yeah. But that's a good question, so. I mean, that's a question that I also had, but, yeah, it's a 5' to 3' direction is the one that it will do.